Hi everyone, I will present my recent work about adenovirus detection environmental wastewater samples with a portable optic fiber sensor platform. When we look at the motivation for this work, it is known that human adenovirus circulate widely and infect millions of people worldwide. It has different infections like gastroenteritis, respiratory infections, or eye infections, and we know that it can be very harmful, especially for young children and babies. So detecting this virus is important, we know that, especially for virus detection. Generally, cell culturing is used, but these kind of viruses growing very slowly, so this cell culture detection takes at least 2 weeks. It is too long. With this objective, we aim to develop a portable optical fiber sensor for on-site detection, because in environmental samples, the on-site point is very important. So, for example, in medical applications, a human come to your medical center and you can take the sample here. But for environmental applications, we need to carry our system near to the environment, especially in living cell detections. Time is very important, so we need to detect this sample right away. So the on-site is a key point. Here we use the antibody-based direct detection for fast and easy to use purpose. A highly sensitive detection developed with using adenovirus specific hexon antibody. When we look at adenoviruses, they are waterborne virus. Only human enteric viruses contains double-strand DNA. Their size is very small, and they are non-envelope. It means that they don't have an outer lipid layer. Their proteins, you look in the upper figure, is just located in the outer surface of the viruses. So with targeting these proteins, we can detect the viruses very easily. So here we are interested in this hexon protein. And the minimum ratio of the hexon antibody to the adenovirus is 240 antibody molecules per virus particle. This ratio is very important. I will explain later in my sensing mechanism. When we look at the current detection methods and their limitations, as I said before, the cell culture method needs at least 2 weeks. A fluorescent activated cell sorting assay, this assay is very sensitive. They can detect at least 1 PFU per ml virus, but we need 3 days. PCR assays is quite rapid and sensitive, but they are complex detection methods, and they need some pre-experiment such as DNA extraction steps. So at this point, when we look our portable optic fiber sensor platform, as you see in the bottom figure, it is quite simple, compact, and portable. We have all these parts in the one single box, and we can carry this everywhere. We have a sample cell, and there is an optical fiber in it. And also we have a laser source, a detector, and also a computer part in it to control all this system. When we look at the sensing mechanism, when the light coming from the laser to the optic fiber, a very tiny electromagnetic field regenerates, and this wave interacts with the surface-bound fluorescent labeled analyte. The excitation flow force generates, and the amplifier detects this flow force, and we can see directly this in our computer screens. When we look at our sensing mechanism, first we immobilize our hexon specific antibodies directly to the optic fiber surface. Here we use the covalent binding, and then we pump our adenoviruses to the sample cell. As I said before, the ratio for the antibody and the virus is nearly 250, and we believe that this ratio is enough to capture the adenoviruses from the flowing sample. And here we let a little time for the adenoviruses, that all of them come to the sensor surface and bind to the antibodies. And after that, we inject our fluorescent labeled secondary antibody. They are specific for the hexon antibody, and with using this fluorescent dye, we can detect the fluorescent signal. And this fluorescent signal depends on the adenovirus concentration in the beginning. There is an inverse ratio between the fluorescent intensity and adenovirus amount, because when we have higher concentration of adenovirus, we will have less open places for secondary antibody, and the fluorescent intensity will be decreased. And after this signal detection, we regenerate our surface with using low concentration of sodium hydroxide. In the picture, you can see a sample signal profile. After we inject our fluorescent dye labeled secondary antibody, the fluorescent intensity starts to increase. And after a time, we can see this plateau, and we thought that our detection is finished. Then we regenerate our surface and wash the surface. After this PBS washing, as you see, we can get nearly the baseline. So we can use the optic fiber for another sensing. And we can calculate our percentage of signal depends on the control experiment, which has just 10 microgram per ml secondary antibody without any virus. First, we make some optimization experiment. One of them is secondary antibody concentration and binding time optimization. Here we use 1, 5, and 10 microgram per ml secondary antibody, and we let them to bind the surface for a while. And we saw that after 10 minutes, we will have the stable signal, and we conclude that 10 microgram, the highest concentration, and also 10 minutes is optimum. And we try to optimize the sensor surface stability. As you see in the figure, after 5 times, our percentage of signal start to decrease. So we conclude that we can use just one fiber just 5 times. After 5 times, we don't have enough reliable results. After this optimization, we make some dose response measurements, and we tried 2 different times, 10 minutes and 40 minutes. As you can see in the figure, with 40 minutes binding, we have 40 minutes detection, we have much more bindings. And when we look the logarithmic curves, with 40 minutes, we have much more binding. And also in 37 degree, we have the highest binding with the adenoviruses and hexon antibodies. And the linearity is nearly the same for all conditions, and the standard deviation are acceptable for the room temperature, but 37 degree, as you can see, the standard deviation is quite high. Is it because we cannot heat our whole system in 37 degree? I couldn't find this problem for now. I put off the optic fiber from the system and put this to 37 degree with adenovirus solution. Then I put back this virus to this, the optic fiber to the system. So this taking off and putting back make a little make the standard deviation result a little high. So still I try to solve this problem. After this dose response curve, we make a specific experiment to using rotavirus and 2 different bacteria strains, E. coli and Salmonella. As you can see in the figure that with direct detection sensing mechanism, we still have 20% false signal with other pathogen strains. Actually, we believe that most of the false results is coming from just non-specific adsorption in the surface. Also, with this kind of pathogen detection systems, the 20% of false results can be acceptable. In the end of this work, we make spiked environmental wastewater samples application to our system. Here we get different effluent samples from US. So we know that these samples don't have any adenoviruses. So first we spike them, then we try to detect them. And the recovery of all measurement samples is between 95 to 117%, and still we have 20% of false results. But when we look at the coefficient of variation result, it is quite good. And after all these results, in conclusion, we developed a rapid, selective, easy to use, and portable system. And our low detection limit is 100 PFU per ml, and we can use just one fiber to 5 times. And also we can apply this system to real wastewater samples. And we believe that this biosensor developed here could be rapidly used for on-site monitoring, such as other pathogens in environmental matrices, with employing of different probes, I mean using different kind of recognition elements. At the end of my presentation, I would like to thank my advisor, my group members, and also I would like to thank the Republic of Turkey to supporting me in my PhD education. And thank you for your attention.